Good afternoon, everybody. I hope everyone is doing great. Today, I am going to speak about CDISC. CDISC is the standards which are followed by the pharmaceutical industry in clinic and in clinical research organization. Now, why this standard? What is the importance of the standards? We will be discussing this and questions and answering them. Going backward in time, the data are collected properly but not accurately. Let's take an example, a simple one, that is gender. Now for gender, we have only two values and one column name as gender. But with the different studies, it is used to change, like the column used to be given for one study as gender. However, for another study, it was as sex. It was changing from study to study. Not only column names, the values also changed. Like for one study, it was M and F, or it was male and female. For another study, it was 0 or 1, or it was 1 and 2. Because of it, it got difficult to work for the huge data and linking different studies, and it used to take more time for programmers to work with these data. Now, during late 60s, during late 90s, a group of individuals started working together and decided to tackle the challenge of defining a standard across the clinical research. CDISC was formed with a mission to develop and support global platform-independent data standards that enable information system interoperability to improve medical research. Now let's run over today's agenda. We will be discussing about what CDISC is and why these standards are being used. We'll discuss about different CDISC standards and models. We'll be also discussing why are they so important, what are their rules. We'll also discuss the collaborations made by CDISC and different standards which are in progress. We'll understand the term BRIDG and what is the importance of CDISC. And in the end, we'll discuss how SAS is supporting to work on this data with the help of CDISC standards. Let's begin where I left off, that is, understanding about the CDISC. Now, CDISC stands for Clinical Data Interchange Standards Consortium. It is standard developing organization which is dealing with medical research data linked with healthcare. They are developing these standards to enable the computer systems or softwares to exchange and make use of information so that they can improve medical research and related health areas of healthcare. Now, these standards support the healthcare industries from the creation of protocol till the analysis of data and creating the required reports for the submission to the regulatory authorities. That is, they support the accusation, exchange, submission, and archive of data, medical, and biopharmaceutical product development. The consortium releases guidelines and requirements that influence the FDA and PMDA on standards for both clinical and non-clinical data also. CDISC uses control terminology to unify the data collected across the submission. Because of these CDISC standards, they have shown a decrease in resources that is needed by 60% overall and 70 to 90% in startup stages when they are implementing at the beginning of the research process. Now, why does this CDISC matters? With the proper implementation of these CDISC standards, we can increase time and cost involved during the drug development stages. It helps us to reach the goal much faster than awaiting for the data to be analyzed and taking more time and cost. On October 1st, 2016, the Japanese PMDA switched to CDISC standards, and now they require all the submissions filed on or after the date to be followed by the CDISC compliant formats. Not only with PMDA, even FDA had also made the decision. FDA, the US regulatory authority, they require all the submission in CDISC compliant format on or after December 17th of 2016. They have also asked the sponsors and the stakeholders to actively transist their data into CDISC formats before the required date of December 17th, 2016. Now, all the regulatory authorities are making mandatory to submit the requirement in CDISC formats. There are also three major standards which are being followed by the SAS programmers in the clinical industry, which are CEN, SDTM, and ADAM. Now, CEN is associated with the data collection from the non-clinical study, that is, the data while it is collected during the animal trial. Then we have the SDTM and ADAM, which is associated with the data collection from the clinical trials, that is, the drug trials which are being conducted on human beings. We have many benefits of CDISC standards, like we can foster efficiency of the data. With the help of these standards, we will be able to understand the data in much more detailed manner. It will help the healthcare industry to make the process faster and check whether the drug is working accurately or the drug has any side effects which can lead to discomfort for the patients. In each stages of the clinical trials, we'll be able to identify the in-depth information of why the patient's body is reacting to the drug and how it is reacting. The different test results will help us to identify the cure for disease, or the other way round, that is, whether where the standards will help to analyze it more accurately or foster the efficiency. We'll be able to identify from where the data has been collected and how it is being processed. That is, we will be able to identify the complete traceability of the data. Now, while converting the CRF to SDTM format, or while converting the SDTM to ADAM model, we'll be able to trace back from where and how it has been created. Now, this is important part of the process. Now, say for simple example, I have a data which contains more than 40 columns, and from this column, I have removed some of which are not required in one table, and I have created some columns in another table. So here I am categorizing the data according to my requirement, and I have created plenty of tables. And once these different tables are created, I am going to use different tables for analysis. After creating the analysis tables, or during the analysis creation of analysis tables, where I might add a new added columns also. I might calculate the new added columns, or I might also add it during the categorization of the data. Now I will be able to remember when and where I have created. Why? Because I here began only with one table. Now what if I have more than three or four tables which we are going to work with? I won't be able to remember where the column came from, right? I won't be able to trace them from where it has arrived in the reports and which I am going to create. And the derived columns, whether it is correct or is there any missing values in it, I will be able to judge or identify those. That is where there are many more questions arises here. Now this is the reason why traceability is more important. Now if I am going to follow a specific standard, there is more room for improvement. We usually think that standards will limit ourselves to follow only those that is shown or to be done in the requirements, as per the requirements or the standards. But what if I have a new variable which I cannot include and it is not being mentioned in any standards or the tables to be created? That is where among the CDISC standards they have a bit leniency. I can create new tables with new name, but it should be abbreviated with two letters and it should begin with either X, Y or Z. Here they are giving us the scope of creating something new, and this is where CDISC evolve the standards and or change these standards accordingly and release them in newer version. This is how CDISC is enhancing and the innovation and improving the standards day by day. Now, CDISC focus also on data quality too. That is where we are collecting the data and analyzing them, and it should have a meaning, or I can say it should have the information that can lead us to proper insight and accurate data driven decision. To get a proper data driven decision, the data should be perfect. That is, these standards support us with the help of CDASH. Now, this standard supports to develop the CRF, that is case reporting form. And always remember, if we do not have a good data quality, will not be able to get a quality insight which we require. Hence, the quality of the data is directly impacting on the statistical outputs which are generated according to the statistical analysis plan. Therefore, the data which we are collecting should be clear, understandable, and precise, which will in turn help the medical investigators to provide accurate data. Next one, we have data sharing. When we complete the analysis and submit it to the regulatory authorities and get the approval. We publish these reports and data. Now here I am not following a specific standard or the other scholars or researchers will find it difficult to understand it at that time. Also as those reports which are created is in my own format, and it will be also difficult for them to correlate with other studies too which they are researching for. But if we have a unified standard, it not only becomes easier for the scholars and researchers but also for 20 other people and other purposes too. The standards is helping in reducing costs also. Here the question arises, how can a standard help in reducing the costs, right? Now let's say I am the owner of the pharmaceutical company who is trying to release a new drug for COVID-19, and I am not going to follow any standards here. So I will be recruiting the manpower to do the work, collecting the data. I have scientists, I have the people who will do analysis and creating the reports and so on. Not only with the people which I am going to recruit, I need a proper place to store the data. So I need the people also who will have this, who can read this data and understand and create the different required reports. Now, I don't have a proper plan here to make sure that all the data is collected properly, or the analysis is done properly, or the data transfer is done properly. But what if I follow the standards here? I know how many people can be given a task for each department. I will be able to plan out the cost involved and also can reduce if I don't require that part of process which I might include it in the study or I might not include it in the study. I would not be able to only reduce the cost but also time here. And clubbing with the reducing cost part, I will be also having a streamlined process here. I will have a proper plan which I will be able to execute it, and if there is any deviation, I can bring back the process to the required output if I know and I am able to identify it where it is going wrong. Now I would be able to work on this. And last not the least, I can have increased chances of predictability. With the standard, I will be able to predict the future with the data, and I will have that whether the drug is which is administered can be giving the cure, and it can also or it can create any different side effects for any human beings, whether that person is having any disease prior to which whether we are working with. So if it is going to work with a smaller group, it will work also for the larger group too. Now over the last decades, a number of models have been developed also with CDISC to support the different needs of the clinical trial data. Now the table here shows the list of different more critical models where the CDISC has helped over the years and their purpose too. Now we have standard exchange for non-clinical data that is abbreviated as SEND. Now this is the one which is required standards for data submission to FDA. SEND is implementation of standards which specifies a way to present non-clinical data in a consistent format. Now, SEND implementation guide is the document which provides the implementers with specification for implementing SEND, including how to model various non-clinical endpoints, rules to doing so, and examples with sample data. The non-clinical, the main focus is on the individual endpoint data here. Now, endpoints are typically to map the domains, essentially the data sets with a number of columns, and how the cell is used for non-clinical trials. Now, there are numerous steps used for non-clinical trials to get a better picture of all the different phases. Now in this picture we can see that they have do this by planning the complete study, then collecting the required data, organizing them according to the requirement and analyzing them to understand how the drug has affected the patient who are injected with different drugs. Now let us understand one by one. Protocol representation is abbreviated as PRM. Now, planning is the process of thinking about the activities required to achieve the desired goal, and it is increasing the efficiency and reduces more risk. So here, now to plan the clinical trials, which includes the process, the people involved in it, we use PRM. But PRM is not just a specific template, rather it is a template which is designed to meet the purpose of a given organization or a study type. The PRM elements are developed so that the protocol information could be reused and repurposed across the multiple documentations database and also the systems. There are PRM elements which have been found to typically cross the study protocol, and these elements are related to four major areas of the clinical trial or study registry. We have elements related to eligibility criteria. We have two parts of the study design. That is, for the study design part 1, it includes the elements which are related to the experimental designs, such as arms and epoch. And the last one, we have the study design part 2, which includes the elements related to study schedule of events and also activities. The other model which we use is CDASH, that is Clinical Data Acquisition Standards Harmonization, which is abbreviated as CDASH. Now, Clinical Data Acquisition Standard Harmonization, it establishes the standard way of collecting data consistently across the study and sponsors. It helps in collection of data formats and also the structures which are provided with clear traceability of submission into SDTM. It helps in deriving more transparency to regulators and others who are conducting data review. Now, next we have Steady Data Tabulation Model, which is abbreviated as SDTM. It provides a standards for organizing and formatting data to streamline process in collection, management, analysis, and reporting. Implementing the SDTM standards support the numerous factors, the factors which are an aggregation and warehousing. It fosters the mining and reusing. It facilitates sharing. It helps to perform due diligence and other important data review activities. And last one, not the least, it improves the regulatory review and approval process. Now, SDTM is one of the required standards of data submission to FDA US and PMDA Japan. To implement the SDTM standards, we use SDTM IG. So it is nothing but the implementation guide which helps us to know what the different column names to be created, what is the type of column, what are the different tables to be created. It also has the guidelines for creating new table which the standards does not include. Not only with that, we have the last model, ADAM model. That is, it is analysis data model, which is abbreviated as ADAM. Now here, ADAM supports efficient generation, replication, and analysis result of the analysis data and data representation of SDTM. ADAM is also one of the required standards of data submission to FDA US and PMDA Japan. Now to implement the ADAM standards, ADAM IG is used. The ADAM IG is the specifications of ADAM standard table structures and columns, including the naming convention, and the ADAM standards has been developed to meet the needs of FDA and the industry. It provides the standards for transferring the datasets between sponsors and CROs, that is, the contract research organizations, development partners, and also independent data monitoring committees. Now in this picture, we can also see the part which includes the data exchange. Now what is this data exchange? Data exchange is the standards which are to optimize and represent the serious content. It facilitates the sharing of structures, data across the different information systems. Operational data model, that is abbreviated as ODM.xml. It is a vendor neutral and platform independent format for exchanging the data. It also archives clinical and transactional research data, along with their associated metadata, administrative data, reference data, and audit information. It facilitates regulatory compliant acquisition. Then we have the defined.xml, which transcripts the metadata, describes any tabular data set structure. Now, this defined.xml is required by FDA and PMDA for every study in each electronic submission to inform the regulators which tables, columns, control terms, and other specified metadata are used. When when used with CDISC foundational standards, it provides the metadata for human and animal model table also, using both the SDTM or the SEN standards and also the analysis table using the ADAM standards. Now we have data.xml, which supports the exchange of tables that are being used database on the define.xml metadata. It balances the define.xml and provides alternative to SAS support transport format for the exchange of steady dataset. And the clinical standards in RDF, it provides a representation for the CDISC foundational standards in a model based on research description framework, that is the RDF. And RDF provides executable, machine readable CDISC standards from the CDISC library. Now, collaboration is the process of two or more people or organizations working together to complete a task or achieve a goal. It is similar to cooperation. In other words, collaboration is the purpose of a purposeful relationship in which all the parties strategically choose to cooperate in order to accomplish a shared income. However, let's understand how CDISC is collaborating and improving the standards of healthcare and clinical research industry. Now, a new project was launched named CDISC 360. The committee is working on this project and it is increasingly steady. Then we have the project's determination to innovate the data structure, clinical data structures as the concept-based standards to store this CDISC library. This new standards paradigm will enable the automated development of specification and also automation generation. There are three cases which are identified as the proof of concept for concept-based structures. The first one we have as identifying as the we have as defined. In this case, it is selecting the standard concepts needed for a study from CDISC library as linked metadata. It is based on an identified study endpoint. The second one, we have it as built. Here, they configure the selected standard concept for the study design and also the auto-generated study for the artifacts. The last one is executing. That is here they are auto-populating the study datasets and the TLFs which are being content and which is driven by the configured study metadata. Now this project have more numbers. Over we have 100 participants for 36 members. Also we have different other various things which are being collaborated. That is, apart from the CDISC 360, they are also working with real world data content. Here, they are trying to embed and they are trying to collect the data on the basis of real world. So they will be using different monitors, like they will be using different variables, wherein they will be able to identify the heart rate and also different other technologies which will be new coming in. And due to this COVID pandemic, there are different other standards being developed. They are using the Delphi method. Now, Delphi method is nothing but where they are coming in together, where all the experts are coming in together, having a discussion, and they are trying to bring a solution to the problem. And with this they are also supporting different community too. That is, they are able to support the different research community there. Now here we are also having another model which is the standard for biomedical research concepts, that is the BRIDG. Now, this model, it is very useful for in plenty of places. That is, we are able to get different reference model. With this model bridge, uh, we are able to also use data integration or mapping those solutions. And also, we are able to give in a proper exchange format. Not only with this, they are helping out with the different physical data, and wherein they will be able to identify those. Now, for each harmonization of the CDISC standard with the bridge, there is a worksheet in the bridge mapping sheet spreadsheet, and it contains different details and mapping tags for each of these CDISC elements are defined on this corresponding bridge elements for it the model itself. So the CDISC standards are harmonized with the bridge, and it includes bridge 3.5. We have statistical version 1.0. We have CDISC lab model version 1.0.1. We have C-dash version 1.2, and also we have trial design model. Now, till now, we were able to understand about CDISC and the different standards of CDISC. We have also understood what are the collaborations made by these CDISC. Now let us see what is the role of SAS and how are they supporting the clinical research industry. Data standards are critical component in the quest to improve the global public health. In the inefficiency of this collection process and the analysis of this patient or the health related information drive up the cost of this drug development for life science companies, and it is affecting negatively not only with cost but also the quality of healthcare delivery of the patients and the consumers. Now, SAS provides a solution to make use of these data standards, such as those from the CDISC, and to provide them more with effective and efficient use of medical information by all the members of healthcare and the life science ecosystem. With a strong commitment, they are developing and also implementing the data standards in life science. And SAS has been a member and an enthusiastic supporter for the CDISC since 2000. These are the different ways how SAS is involving with the CDISC. They are participating in the CDISC industry advisory board meetings and making decision makings. They have involved with the CDISC data model team for ODM, ADAM, protocol and representation group. Not only with that, they are also with the trial design model too. They have also participated in strategic serious development initiations such as BRIDG model. They have involved themselves with numerous other demonstrations in software like connectathons. SAS has also different collaboration in the CDISC interchange and other projects to explore the interoperability of the CDISC data model, and also they support the CDISC training and implementation activities. In addition to the helping of defined CDISC standards, SAS is making certain that our products and the solution support the implementation of the CDISC data standards. This Here they are using this clinical SAS toolkit which provides the support of clinical data standards and base SAS. Not only with that, the clinical CDISC standards such as SETM can be effectively implemented in the solutions like we have the SAS Life Science Analytics Framework and also this clinical data integration. Now, today we have discussed a lot about CDISC and the different standards used by the CDISC. We were able to understand the different collaboration made by the CDISC during the COVID pandemic. We were able to see different models. That is, we were able to see the SCTM, SEN, PRM, C-dash, and Adam. And in the end, we were also able to see how SAS is evolving in to help the CROs and the healthcare industries by working together with the CDISC. Now here I will be ending my webinar by thanking everyone for spending your time for listening to me, and I hope everyone will join for the upcoming webinars. Thank you again once again and have a great day. Bye.